Hello everyone, and welcome to Chapter 7, which is going to cover biotechnology. So happy you could join me for this lecture. Let's go ahead and get started. So here are our learning objectives. I'm sure you can read them yourself, but here they are. So jumping in, what is biotechnology? What does it promise as well? So understanding a phenomenon, as we've discussed prior to this, is really cool, and it's pretty exciting when you make that type of discovery. But what feels even more satisfying when it comes to being in the sciences is being able to control that phenomenon. And that is what biotechnology and genetic engineering both help the science community do. So biotechnology is the use of technology to modify organisms, cells, and their molecules to achieve practical benefits. A few examples of these are like tests for cancer through your saliva, being able to regenerate neurons, so those are those nerve cells in your brain and spinal cord. Genetic engineering is going to be the manipulation of organisms' genetic material by adding, deleting, or transplanting genes from one organism to another. We'll talk about how that's been done. But a few ways that we currently use biotechnology, like very widespread, there are 3 examples I want you to know. The first is agriculture. So we have made plants, and more so specific is crops like corn and soybeans and wheat and various things like that, to be resistant to pests as well as diseases and potentially environmental factors as well. The next is human health. Obviously, we've come really far with our advancement of medicine and prolonging life in humans. So this would be like treating diseases. Good couple of examples are type 1 diabetes. That would have been a death sentence without modern technology and biotechnology. And then anemia as well, so a lack of red blood cells, and sometimes that's genetic and sometimes that can be caused by other factors, but we do have ways to combat that. And the third is forensics. That has come a long way in a short period of time thanks to the use of biotechnology. So this would be an example of like matching DNA when it comes to crimes. So a few important processes that underlie many biotechnology applications include these 4 steps, and not all of the tools are used in biotechnology applications. Some use only one or a few of these techniques, but we are going to talk about each one of these individually. So we have chop, amplify, insert, and grow, and most of these have everything to do with DNA or inserting DNA into something else. And we'll talk about them each individually. Where this comes into an applicable thing, so a couple questions to kind of think about to get you in the mindset of why we would want to use any of these tools in biotechnology is how would you create a plant resistant to being eaten by insects but make it to where it's still able to thrive in its environment, or a colony of bacteria that can create human insulin and thus be harvested. So, these are kind of ways that we are able to do those various processes. So this first one is chop, and you will notice if you kind of read through this very briefly as I am speaking, that it is identical to our lab 5 of the whodunit / murder mystery. So this is all an example of that method that we used to chop up our DNA into fragments in order to figure out who murdered Mr. Body. In this example, they are using a restriction enzyme, not one that we used, to cut sequences at the ATCGAT, which, remember, that is a DNA palindrome. So obviously it's not a literary palindrome, it's not the same forward as it is backwards, but it is the same going forwards on one side of the DNA as it is on the other side of the DNA. So that's a very important thing to kind of make a note of here. So, as we've kind of figured out, chopping is going to be isolating a gene of interest using our restriction enzyme. And these restriction enzymes can chop up sequences, base pair sequences that can be 4 to 8 base pairs long. It doesn't have to be the same length that we used in lab. And there are dozens of restriction enzymes that each cut at specific locations with specific sequences. They're not all the same, as you can see from the example we did from lab versus the example that we have here in this illustration. From there, the next method is going to be to amplify. This is using a polymerase chain reaction, or a PCR, and the purpose of this is just to duplicate a tiny piece of DNA repeatedly using heating and cooling. And as you can see with this little example, they are heating and cooling, which is breaking apart our nucleotides and then putting them back together, essentially. And then the result is going to be 2 identical copies of the original segment of DNA. This process can be repeated again and again until there are billions of identical copies of the targeted sequence, which is honestly pretty cool, but that's a PCR. The next method is to insert, which is using plasmids, which plasmids are just circular pieces of DNA to transfer DNA from one organism to another. And that final product is going to be known as a transgenic organism. So it's sort of a Frankenstein organism, if you will. So sometimes, instead of the example that we have up here using bacteria, we can use viruses, and these are used to infect organisms and transfer the genes that we are wanting transferred that have been implanted into that virus, which is honestly like the coolest thing ever and also slightly terrifying. The last method is to grow. So this is creating a gene library. Essentially, all you really need to get from this is that they are DNA fragment clones. We'll talk about what you probably think of when you think of the term clone a little bit later, but that's all this is. It's just creating essentially DNA fragment clones. The next item we're going to talk about is one of the coolest things you're probably ever going to hear about when it comes to biotechnology advancements, and that is CRISPR, which is an acronym standing for Clustered Regularly Interspaced Short Palindromic Repeats. Try saying that 5 times fast, I bet you can't. But this is at the cutting edge of biotechnology. It's a tool with the potential to revolutionize medicine. We're going to talk about what it does. So essentially, it is a genetic Photoshop. And what I mean by that is it's a system for editing DNA with a great deal of precision and efficiency, kind of better than you've ever imagined could be done. And we're going to talk about how it works and some of the drawbacks of it. But what this does is it's going to enable researchers to modify almost any gene in any organism. And that's going to be like bad genes, especially, which is absolutely the coolest thing. Imagine, being born with this genetic condition and being able to, genetically Photoshop that bad gene out. It would be amazing. So here's how it works. CRISPR is a gene editing system that brings greater precision and efficiency to modifying genomes. It's going to be able to identify a particular DNA sequence of interest and be very specific about it too, which is kind of cool, where researchers can synthesize an RNA guide molecule with a sequence that matches the target gene to be sliced. From there, the sequence for the CRISPR RNA and Cas9 enzyme, so that's just a little enzyme there, are introduced to target cells using a plasmid. Remember, that's just the round DNA. And then from there, within the cell, the RNA leads to the Cas9 enzyme to exactly the desired location on the DNA, and Cas9 cuts the DNA out of there, just absolutely photoshops out that blemish, if you will. And then finally, at the location where the DNA is cut, a sequence can be inserted that repairs or alters the host cell's DNA, which, again, you're just photoshopping in what you want to be there genetically. So cool. So the potentials of CRISPR are like uncountable. It is absolutely just the most amazing, breakthrough technology because of its ability to snip DNA precisely at a specific sequence. So a couple examples that we have of how CRISPR can really just make such a difference is, for example, mosquito DNA can be altered to block malaria, which malaria is a little parasite that gets into your red blood cells and basically destroys them as they reproduce inside of your red blood cells, which then causes an immune response when they all burst out and go crazy in your bloodstream. And then they hide inside the red blood cells that are remaining. But by doing that, it causes anemia, which can lead to death because you don't have any red blood cells to carry oxygen throughout your body. So if you can modify mosquito DNA to where it can't transmit malaria from one person to another as they are feeding on blood, that would be a life-changing or world-changing experience, because that is one of the leading diseases that is causing such a problem in Africa, in other countries as well, but especially Africa. The next are human trials being underway in which CRISPR altered white blood cells that are being used to fight cancer cells. Again, kind of hitting home with these really big world-changing things, a cure for cancer. Can you imagine that? It would be absolutely astounding. And then a little less interesting, I suppose, but it's still kind of cool. Beagles were engineered to be born with twice as much muscle as a typical dog, which, maybe not as cool as, curing cancer and getting rid of malaria, but still pretty neat. Now, with every amazing sounding thing, kind of like a Billy Mays commercial, or, one of those really crazy commercials that's promising you, complete 20 pounds of weight loss in 1 week, there are drawbacks. As far as CRISPR goes, most of them have to do with more of a public eye kind of issue rather than an actual issue with CRISPR itself. So the big one is legal issues surrounding who invented it and who can profit from it. There's various very prestigious schools that have fought and continue to fight over the patent for CRISPR. So who is going to make the most money off of this really amazing product is one of the hang-ups with it, which sounds so asinine, but hey, someone's got to profit, right? The next is ethical issues. So one of the cool things about CRISPR is essentially it leads way for the potential of editing human embryos, which I've always considered the term Build-A-Baby as like describing that. So where that comes into an issue is very ethical, whether or not it is okay or our right, or are we playing God essentially in editing human embryos and building an infant, building a baby, to your exact description. You could say that you wanted your kid to have blue eyes, blonde hair, small nose, big teeth, whatever, have your freckles or moles that you want them to have, whatever it is. but essentially it's build a baby, and there's ethical issues behind that. The other ethical problem is the potential for editing germline cells. These include sperm and egg cells, because gene changes can be passed to the next generation. Who's to say that this, baby that you built, not created, you built it, then passes on that genetic information that you biotechnically created in it. That information is going to be passed on and put into the human population, if you will. So there is an ethical issue in putting something, kind of weird to say man-made, because, obviously babies are man-made, but you get what I'm saying. Lab created. You are putting something lab created into the genetic pool of our human population. And then the last risk of CRISPR is it's difficult to predict consequences of introducing altered genes into genomes of natural populations. And the best way I can describe what this risk includes is the butterfly effect. If we take out mosquitoes' genetic ability to pass on malaria, what will that then cause? What flap of the mosquito wings will cause what city to fall, if you will. Kind of a goofy way to say that, but hopefully you get what I'm saying. So moving on to biotechnology being able to improve food nutrition and making farming more efficient and eco-friendly. As maybe you've potentially heard, GMO products is what we're starting to talk about here. And honestly, we're going to talk about how GMO isn't quite as scary as it sounds. If you've been on the fence about GMO products, or if you just don't care at all, that's okay. It is important to do research, and of course you should always, question what you hear. Typically I'll always try to steer you in the right direction, but who knows? Maybe by the time you hear this, information will have changed. So we have this term genetic engineering. That is the manipulation of a species' genome in ways that do not normally occur in nature. So, for example, we have this picture here of ancestral corn, which looks absolutely nothing like our modern corn. We have genetically engineered corn that we eat today to look and taste and grow the way that it does. Ancestral corn was pretty much just inedible, or at least really, really difficult to make edible. We have now made corn one of the largest worldwide crops, aside from maybe soybeans and wheat. Next is recombinant DNA technology, which has sped up that whole process of genetic engineering, which honestly humans have been doing for thousands of years, but very rudimentary. Recombinant DNA technology has basically taken that process and made it just accelerate so much, to the point where, we could come up with a new genetically modified product within a few weeks, maybe a few months. So kind of cool. An example of what we have done to genetically modify plants, and it's mostly for good purposes. Obviously, we wouldn't necessarily try to do this with nefarious purposes, I hope. For the most part, it's to be cost efficient and environmentally friendly, as well as to be good to, the people of the world. And this is a good example of that last little feature there. So 10% of the world's population actually suffers from a vitamin A deficiency. And in order to make vitamin A, you use something called beta-carotene, which is found commonly in carrots, as it does sound like carrot. So beta-carotene. It is what gives carrots its orange color, which is kind of cool. So essentially, as mammals, we take beta carotene and then we convert it into vitamin A, which we then use in various ways. So what scientists did is they modified white rice and basically inserted this DNA from daffodil genes, which is that beta carotene, as well as some bacteria genes, which then causes something called golden rice, which is that product of adding that DNA to white rice, which then adds beta carotene into that white rice, which then puts vitamin A back into the diet of many, many people, because most of the world eats rice, actually. Kind of cool. Really cool way of using, you know, modified crops to better the world, if you will. Almost everyone in the United States uses genetically modified crops regularly, whether they know it or not. Honestly, I kind of consider everything that we have today to be genetically modified, because there's very little that hasn't changed since humans started cultivating and using agriculture more commonly. Same goes for animals. If you ever want to see what your dog potentially should have looked like, you know, many, many, many years ago, look at dire wolves and larger wolves in general. Your dog is definitely not, you know, naturally occurring. And it doesn't matter what kind of dog you have unless you have a wolf. Wolves were domesticated, which, you know, in a way you can kind of think of it as being genetically modified. Maybe not on purpose, which most of these crops that we've talked about and will talk about were modified not on purpose through the years. You know, as I said, genetic engineering was done for thousands of years. We're only now sort of more specifically using recombinant DNA technology to change things at a faster rate than we had before. So two factors driving adoption of modified crops in the U.S. include plants with insecticides engineered into them and plants with herbicides, herbicide-resistant genes engineered into them. So these are kind of interesting factors because insecticides obviously are going to keep insects from consuming these products, these crops, and then herbicide-resistant genes are going to help with crops being treated with herbicides in order to kill the weeds out of the fields. So what this does is it reduces the cost of producing crops. It also lessens the topsoil erosion, which is and has become a very big factor in a lot of agricultural problems. If you think about the Dust Bowl, I think during the 20s, I could have that wrong, I'm not a not a history major, but the Dust Bowl was terrible because there was absolutely nothing holding that topsoil in place. There were no plants because those fields had been over used. So it caused these really intense dust storms that, you know, suffocated people. It was terrible. So how we get some of these engineered insecticides into our plants actually started from bacteria. So this bacteria, Bacillus, and I'm gonna try this word, thuringiensis, sure, we'll just call it BT, produced crystals that were basically just natural pesticides, which is so cool. So what scientists did is they took this BT crystal gene and inserted it into corn, which made it to where they have this natural pesticide that repels insects and, you know, various pests like that, which, It sounds really, really cool if you think about it, and it's natural. I mean, not that natural versus unnatural is a good or a bad thing, and we'll talk about that later. So herbicide resistance was also started by a bacteria DNA, making it possible to be inserted into those plants and making the plants resistant, which made it to where the farmers were able to spread herbicides and not harm their crops, but able to kill the weeds of their fields. So it's not just plants that we have genetically modified. We also have modified animals, especially animals that are used in farming. Some of this can be good, some of this can be kind of bad, and really the best way I can really encourage you, if these things bother you, is to put your money where your heart is. If you're feeling sort of a conscience pull towards not wanting to support various things, companies have to listen to where your money is going. And that doesn't mean that those organizations are going to change overnight, but get enough people to stop buying various things, then those companies are going to go under, or they're going to change the way that they produce their products. In this case, we have salmon that have been given a growth hormone gene that is going to keep it growing year-round rather than the way salmon normally grow, which is only in the summer months. So we've got this much larger fish compared to a more natural fish that has not been modified. One of the biggest concerns when it comes to these genetically modified fish is that they potentially could cause an allergic reaction, as well as an environmental concern, being that the breeding of genetically modified fish with wild fish, if that genetically modified fish were to escape captivity and breed with fish in the wild, then those genetically modified fish, which grow year-round instead of just in the summer, would potentially outbreed and outcompete other endangered species, and that could lead to a pretty big environmental issue. And again, with sort of these risks versus rewards and using an animal modification as an example, unfortunately, as silly as this chicken looks, I can't imagine he's too comfortable due to the fact that birds should have feathers. I mean, there's probably a few birds that don't, and they're doing just fine, but chickens were created, I guess, in the world to have feathers naturally, and these chickens were modified to not have feathers. And while that makes it much cheaper for farmers and consumers, because the process of removing the feathers adds a cost as well as time, there are unintended consequences, including vulnerability for the chickens while they're alive to mosquitoes, parasites, as well as sunlight. Because of course their skin is not made to be, you know, exposed directly to sunlight all the time like that. That can't be comfortable. So again, putting your money where your heart is. If this bothers you, making sure to know where your meat or your products are coming from is very important. Do your research. So there are 6 primary concerns about GMOs. We are going to talk about them. The first 3 are on this slide. So the first is that organisms that we want to kill may become invincible, and this actually is a pretty decent concern as far as genetically modified plants. There was a weed-resistant canola plant in Canada that ended up spreading outside of the field that it was intended to be in, and it spread out of control to other fields, and it couldn't be contained because it was weed resistant, it was herbicide resistant, so there was nothing they could do to essentially kill it without harming the field that it was in, and of course the crops that were in that field respectively. The next is organisms that we don't want to kill may be killed inadvertently. I think this example is probably, so close to my heart. Monarch butterflies are actually the potential on that list of organisms that can be killed that you don't want to be killed. With the pesticide bacteria that we talked about before, that was those Bt crystals, the crops that have that gene inside of them produce a pollen that has that Bt crystal gene in it, so that pesticide gene is in the pollen. And if that pollen were to land on milkweed, which milkweed is a monarch butterfly's pretty much like favorite thing in the world, it would kill the monarchs, which this hasn't happened yet in recorded history, but it is hypothesized that it could and potentially will happen, and that would be just devastating if that were to happen, not just to monarchs, but other, potentially pollinators like bees or wasps or, birds or things like that. That wouldn't be good. The third concern is genetically modified crops are not tested or regulated adequately. There are guidelines in place, but the problem is that technology is moving incredibly fast right now to where it's a little bit difficult to keep up with these guidelines and policies. There are still policies in place, things are still being tested and regulated. It's just a little bit hard to do on the scale that technology is moving. The next set of concerns include eating genetically modified foods can be dangerous. This is sort of a catch-22. So in the 90s, a gene from Brazil nuts was transferred to soybeans in order to make the soybeans more nutritious. But what ended up happening is a gene that caused allergy-causing chemicals to be put into soybeans or produced by soybeans sort of snuck in from the Brazil nut. So what had previously not caused allergic reactions from soybeans then started causing allergic reactions. So things like that can happen. Next is a loss of genetic diversity among crop plants being risky. An example of this was from the 1880s, the Irish potato famine. So this is where crops become more vulnerable to environmental changes and pests due to a lack of genetic diversity, which this has happened. It's not good. What this could cause is, potentially a worldwide famine if everything is essentially genetically the same. Then everything is genetically susceptible to being killed off in some capacity, whether that's by disease, environmental changes, or pests. And then the last concern about GMOs is hidden costs may reduce the financial advantages of genetically modified crops. This is sort of more of a concern with greedy companies rather than a problem with GMOs as themselves. So companies will engineer sterility, so the inability to reproduce, into these crops, which then forces farmers to buy new seeds each year, which you might be thinking to yourself, that sounds really awful. It's a way for these companies to make money because they're producing these really great products that are cost efficient in a lot of other ways, so they have to be able to make money in that way. So it sounds really cruddy because it is, but I guess those companies have to make money too. Moving on to medicine, so we're moving away from plants and animals and moving into medicine. So the treatment of diseases and production of medicines are improved with biotechnology in order to prevent diseases, treat diseases, and cure diseases. Now, two of these would be absolutely the most ideal, obviously preventing a disease before it can develop, or curing a disease if it has developed. Those are the two primary positions that we would want to be in thanks to biotechnology. Unfortunately, the tier that we are most likely in and going to stay in for a while is to treat diseases that have already exposed themselves, as well as probably not be able to cure them. So it's just going to be continued treatment. An example of this is the treatment of diabetes, specifically type 1. So where this type comes in is that it is genetic. It is not caused by diet or inactivity. This type of diabetes is where your body cannot produce insulin on its own. So shots are necessary to inject insulin into that person's body so that they can properly digest and absorb the nutrients from their food and properly use those nutrients. So where our biotechnology comes in, insulin produced using recombinant DNA technology is making it to where we can kind of change how people with type 1 diabetes experiences their life, as well as being able to live in general. Because as I said earlier in this lecture, type 1 diabetes was essentially a death sentence. Not being able to produce insulin, I mean, you'd die if you didn't have those shots. Other achievements in biotechnology revolution, one includes the human growth hormone, or HGH. This is a hormone produced by your pituitary gland, which is your master gland in your brain that produces lots of hormones, lots and lots, and essentially insufficient amounts of growth hormone can lead to dwarfism. Too much can lead to gigantism. You can think of Andre the Giant as being someone who had giantism. And now we can produce growth hormone using transgenic bacteria. So remember those transgenic organisms that we were talking about before? We can now basically produce human growth hormone using bacteria, which is super cool. The next hormone is erythropoietin. This is primarily produced by the kidneys, and it is used to create red blood cells. It can now be produced from cells derived from hamster ovaries, which is probably one of the weirdest things you're going to hear today. But erythropoietin is very important in helping treat anemia, because you want to increase the number of red blood cells, and that is what that hormone does. Although one of the issues with erythropoietin is becoming prevalent in the sporting community, where you have blood doping scandals. So that is increasing your amount of red blood cells so that you have more oxygen going to your muscles, etc., etc. There's a few other things that are involved in blood doping, not just erythropoietin, but you've maybe heard of erythropoietin before. It's also called EPO. So I don't know, maybe you know some about it, maybe you don't. It's kind of cool. Continuing with medicine, we have something called gene therapy, and this is where biotechnology can help diagnose and prevent genetic diseases, but has had limited success in curing them. Again, we're kind of treating it or preventing it. Curing has been a much harder venture, as much as we would like that process to, you probably heard a little bell. That's my cat. Move, honey. Had little progress with curing various diseases. So screening for genetic disorders generally focuses on 3 different scenarios. The first scenario is a given set of parents likely to produce a baby with genetic diseases. This essentially just tells the parents how likely it is that their child may have some disease that could be passed from parent to child. So since screening began in 1969, our little example here, the incidence of Tay-Sachs disease has been reduced by more than 75%, which, if you know anything about Tay-Sachs, it's a very terrible disease that destroys nerve cells. So those are the cells inside of your brain as well as your spinal cord. And of course, they make up your nerves as well. But the life expectancy, which normally this is diagnosed in infants around 3 to 6 months old, is that they will live maybe to 5 years. So that is a very horrible disease. I can't imagine being a parent and being told that your child has Tay-Sachs. From there, the second scenario is a baby being born with a genetic disease, if they'll be born with a genetic disease. Examples include cystic fibrosis, sickle cell anemia, Down syndrome, and an increasing list of others, including something called SCID. SCID is an immune system malfunction where the life expectancy, very unfortunately, is only about a year old. So the child in the picture here is actually 12 years old, which is relatively old for a child with SCID. But that is made possible because this child was put into a bubble, essentially, in order to survive. So by doing these genetic screenings, we can see that this child, before it was even born, that it would have this condition. The third scenario is an individual likely to develop a genetic disease later in life. These include things like breast cancer or prostate cancer, skin cancer, and others. So how likely are they potentially to develop these various conditions, which helps with preventative measures such as getting a prostate removed or a mastectomy, depending on how severe it could be. Maybe a very aggressive double mastectomy, which means removing of the breast tissue. So that could be absolutely life-saving for someone if they know how likely it is that they could develop those various conditions. So why has gene therapy had such a poor record of success in curing diseases? Well, first of all, the difficulty getting the working gene into the specific cells where it is needed, difficulty getting the working gene into enough cells at the right rate to have a physiological effect. And for those first two, honestly, I kind of think about how hard it would be if you had just a single thread and then like a million needles and you were going for a very specific needle that you were trying to thread. It would just be so hard to do that, and that's essentially what is happening with this gene therapy. The third is difficulty arising from the transferred gene getting into unintended cells again, getting that thread into one of the needles that you didn't intend for it to go into, and then difficulty regulating gene expression, which we've talked about gene expression in the last lecture. Moving on to cloning, both potentially in the way that you probably imagine what cloning was. So this ranges from cloning genes to cloning organs to cloning whole individuals, which, if you wanted to know, that is called whole organism cloning. That is where you clone the entire genome of an individual. So this would cause an exact copy of that individual phenotypically. Genotypically they're exactly the same, and phenotypically they are going to look exactly the same, but that doesn't mean that they are the same organism. Because, you know, this is where like nature versus nurture comes in. They're going to have different life experiences from the get-go. So they might be an exact genetic copy of one organism, but they are not going to be the same organism. Kind of a weird distinction. And to answer the question on your mind, yes, we absolutely can clone a human, but scientists who have kind of gone into this area of research have all unanimously said that it shouldn't be done. And I'd like to quote Ian Malcolm from Jurassic Park. We don't want to get so preoccupied with whether or not we can that we don't stop to think if we should. And I love that scientists are saying that we shouldn't, because we absolutely shouldn't. I think that would be a very, very big mistake. But hey, that's my opinion. So DNA is an individual identifier. The uses and abuses of DNA fingerprinting can be found with this example. There is this, I don't want to call him a gentleman because he certainly isn't, this creature, this man named Colin Pitchfork, what a name, was the first criminal brought to justice thanks to DNA fingerprinting. What happened was he was a murderer and assaulted two underage girls and left, I guess, deposits of his DNA on the two girls that he assaulted. And another man actually came forward and said that he had committed the crimes, but it didn't quite fit. It didn't quite sound right to the policeman working on it. This scientist, which his name escapes me now, was working on some research that was able to identify the DNA left at the crime scenes, and he was able to look at that and say that both crimes were committed by the same person. So what they did is they crossed the DNA found at the crime scene with DNA of, I think, about 300 different men, to which they eventually found this Colin Pitchfork, who tried to convince a friend of his to basically donate the DNA in his place because he knew that he'd get caught, which he did, and he served 2 life sentences in prison. So a little bit about biotechnology in forensics. Approximately 99.9% of the DNA sequences of 2 individuals are the same, but that still means that there are approximately 3 million base pair differences between 2 people. The exception is identical twins, which are as close to identical as you can possibly get with still having just a few differences between them. And then we have this term STRs, which are short tandem repeats that are used to determine someone's genetic fingerprint, to get a little bit more specific. But that is not to say that these processes are completely foolproof. There are problems that occur with DNA fingerprinting. More specifically, it leads to human error being the culprit, such as mislabeling. While this whole process is very useful for comparing a suspect's DNA to DNA found at a crime scene, it is not 100% accurate every time. So from here, you can go ahead and test your knowledge. I will give a roughly 5 seconds per question. And if you really want to know the answers, you can send me an email. And that is the end of chapter 7. I hope you enjoyed yourself. I know it's not the most exciting thing, but we did talk about some pretty interesting advancements in biotechnology, which is all pretty darn cool if you're into that kind of thing. If not, apologies. I hope you have a wonderful rest of your day. Make sure to get your assignments done and, of course, make good choices.